Hello, everyone. I'm Christy Rizk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is Application of a Novel ISH Approach to Elucidate Splice Variants in Schizophrenia, and our sponsor is Advanced Cell Diagnostics. Our panelists today are Dr. Mohamed Katayesh, Senior Image Analysis Scientist at Advanced Cell Diagnostics, and Larissa Erben, a graduate student in the National Institutes of Health's Graduate Student Partnership Program between the laboratories of Dr. Andres Bonanno and Professor Dr. Andreas Zimmer. You may type in a question at any time during the webinar. You can do this to the control panel, which usually appears on the right side of your screen. Click on the Q&A box in the control panel to ask a question. Please make sure the drop-down menu says All Panelists. If you don't see the Q&A box in the control panel, move your cursor over the main panel until you see the control icons on the bottom. Select the Options menu and then select Q&A. We will ask the panelists questions after the presentations have concluded. Dr. Katayesh, please go ahead.
Thank you, Christy, for the introduction. Hi, everybody. Thank you for joining us today. Let me just share my presentation. So today I'm going to give you a quick introduction about RNAscope technology and an overview about what image analysis methods we use to perform the quantitative analysis in our images. Here is the outline of the introduction. So to start, RNAscope works with a variety of tissue shown here in purple and yellow. The most common of these tissue is formalin-fixed paraffin embedded, known as FFP tissue. The tissue is placed in a glass slide where cells are permeabilized and target RNA is unmasked. This technique works on different kinds of RNA, such as messenger RNA or long non-coding RNA. So after the permeabilizations comes the hybridization step, which includes the hybridization of a pool of about 20 target-specific double-Z probes, shown in yellow and purple here. These double-Z probes target the RNA and allow for amplification of the hybridization signal, as shown in the next step, where a sequential of hybridization of amplifiers and label probes result in detection of single RNA molecules as single dots within a morphological context. These dots can be viewed under a light microscope or a fluorescence microscope, depending on label probe used, and they can be quantified on a cell-by-cell basis. So, more specific details about RNAscope assay. It begins with a unique probe design for detection of RNA. Probes are Z-shaped oligonucleotides, as shown here, composed of 18 to 25 base sequence lower region. This is connected to a 14 base tail sequence via a spacer. When a pair of double-Z probe hybridize next to each other, as shown here, a target binding site, which is approximately 50 base pairs along the target RNA, is formed. The upper region of the 2Z molecules now form about 28 base pre-amplifier region. This is important for signal amplification to occur. For each RNA, a pool of about 20 double-Z target probe pairs spanning about 1,000 bases along the target molecule are designed to specifically hybridize to the target molecule, as shown in the next picture. Now, once the pre-amplifier binding site is formed, signal amplification can take place, and an L-shaped pre-amplifier molecule is added, as shown here, which only bind to the pre-amplifier site. The amplifiers then bind to multiple sides along the pre-amplifier, and finally, the label molecules such as chromogenic and fluorescence bind to numerous sites on the amplifier side, as shown in blue. If one Z probe bind to a non-specific target, it's highly unlikely for the another C probe to bind. Hence, a pre-amplification base or sequence won't form. So this assay is highly specific and provides robust signal amplification with background noise suppression. We also offer BaseScope assays. The BaseScope assay we offer is based on the RNase technology but has specific applications. While RNA is a robust assay to detect gene expression of most RNA targets, BaseScope is specifically used for detection of exon junctions, splice variant, and short target or highly homologous sequences. This is due to a more advanced amplification system that can now allow for a probe as short as 1 ZZ or targeting as few as 50 nucleotides. BaseScope is also capable of single molecule detection and can be used to detect up to 2 targets simultaneously. Next, I'm going to give you a brief overview about how we usually analyze our assay images. So the first method, which is semi-quantitative, shown in this slide, where the number of dots per cell is measured or estimated, and based on this ACD table shown, a score can be assigned to each image. A score of 0 means no staining or less than 1 dot per cell, whereas a score of 4 has greater than 15 dots per cell. This scoring system can be condensed or expanding depending on the level of expression. The second method is based on using more advanced software with the capability of reporting probe copies per cell. For example, a commercial software such as Halo or open source such as MJ or CellProfiler are suitable for that. After having probe copies per cell, results can be plotted in a histogram as shown here with the flexibility to plot, for example, the average dots per cell or per ROI, a certain number of dots per ROI. At the end, once you have this data, you have many options. The third method is H-score or histoscore. This method depends on defining different bins to cover the level of expression, as well as calculating the percentage of cells within each bin. The table on the right shows an example of this method. After you have both information, the data can be presented in a histogram to show the expression level distribution, or can be calculated as a histogram using the formula below. One advantage of this method is that it gives you the dynamic range of expression, or cell-by-cell expression profile, and this can be quantified for the entire tissue section or selected region of interest. I should note that a score can be achieved by semi-quantitative analysis or quantitative image-based software analysis, where the percentage of cells within each bin is known. Also at ACD, we typically use 4-bin system for our calculations as defined in this table. With that, I want to transfer the mic back to Christy and our guest speaker. Thank you.
Thank you, Dr. Kitesh. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We will conduct the Q&A portion of the webinar after the presentations have concluded. And next up is Ms. Erben. Please go ahead.
Hello, everybody. Thanks, Christy, for the kind introduction, and thanks to Mohammed for introducing our listeners to the principles of RNAscope and BaseScope. I subdivided my talk today in 2 parts. In the first part, I'm going to present our published data on how we applied BaseScope to analyze ErbB4 splice variants and identify cell type specific expression. This part will also show how we validated this novel approach. The second section will focus on how in situ hybridization signals can be quantified using CellProfiler. ErbB4 is a receptor tyrosine kinase of the ErbB family. It is activated by EGF-like ligands, which in the brain mainly consists of the neurotrophic factors neuregulins. The neuregulin ErbB4 signaling pathway has been implicated in neurodevelopment and neuroplasticity, both in the peripheral and central nervous system. Importantly, both neuregulin and ErbB4 are at-risk genes for schizophrenia, a neurodevelopmental psychiatric disorder. My presentation will focus on the receptor ErbB4. ErbB4 is expressed at high levels in hippocampal and cortical GABAergic interneurons, but absent from glutamatergic neurons. ErbB4 is also expressed on dopaminergic neurons and oligodendrocytes. 4 ErbB4 isoforms are generated by alternative splicing at 2 loci. Alternative splicing of 2 exons encoding receptor sequences in the juxtamembrane region immediately upstream of the transmembrane region domain known as JMA and JMb make the receptor isolabile or resistant to proteolytic processing by metalloproteases. On the other hand, the second locus resides in the cytoplasmic tail of ErbB4 that is critical for linking receptor binding to downstream activation of signaling pathways. In this case, the SID1 exon is either included or omitted to generate SID1 and SID2 splice variants. Inclusions of SID1 renders the receptor capable of activating the PI3K-AKT pathway. Importantly, expression of ErbB4 isoforms is dysregulated in the postmortem brain of schizophrenia patients. For these reasons, we set out to understand the role of different ErbB4 isoforms in the central nervous system and their potential relevance to schizophrenia. To this end, we initially wanted to understand how the 4 ErbB4 splice variants are distributed in the brain. However, there was a major challenge to achieve this goal with the existing tools because all the exons, JMA, JMb, and SID1 exons are extremely short, ranging between 45 and 75 bases. Most conventional in situ hybridization approaches, such as radioactive and DIG-labeled probes, lack the sensitivity required to detect these short RNA targets. And moreover, in the case of radioactive probes, it is difficult to analyze single-cell localization. This is the reason we resorted to exploring the use of BaseScope. As you heard from Mohammed's presentation, the sensitivity of BaseScope allows for the detection of short RNA sequences. In addition, the chromogeny detection dye used in BaseScope, called FastRed, can also be detected fluorescently in the red channel. In this study, I will discuss, we mostly use fluorescence detection. For the ease of presentation, I have pseudo-colored the red signal to white. The first question we wanted to address in our study were, is the BaseScope assay sufficiently sensitive to detect single exons? And second, are the signals detected specific? To address these points, we designed and hybridized a probe that targets the junction between exon 1 and exon 2. And we call this probe PAN-probe because it detects all ErbB4 isoforms. Using the PAN-probe on hippocampal sections from wild-type mice, I observed a hybridization signal in cells scattered throughout the section and absent in vast majority of cells in the pyramidal and dentate gyrus layers. This was consistent with the expected pattern of GABAergic interneurons. In stark contrast, the same probes on sections from ErbB4 knockout mice missing exon 2 failed to yield signal, with the exception of only a few background dots. Next, as I will cover in detail in the second part, we quantified the signal, and it was dramatically lower in the knockout sections compared to the wild-type sections. Together, this confirmed that indeed BaseScope provides the sensitivity and specificity to detect single exon-exon boundaries in ErbB4 transcripts. Then, we designed probes targeting unique exon junctions for all of the ErbB4 splice variants. And as I showed earlier, there are 4 ErbB4 isoforms, and ErbB4 transcript is either JMA or JMb in the juxtamembrane, and then either SID1 or SID2 in the cytoplasmic domain. Both the qualitative and quantitative analysis in the mouse hippocampus showed that JMb and SID2 are the 2 major ErbB4 isoforms in mouse hippocampus, comprising about 85% and 70% of ErbB4 transcript. Since BaseScope is only considered a semi-quantitative approach, we compared these results with the more conventional quantitative method of Tachman quantitative PCR from isolated RNA. And it was very encouraging to see that these results are similar. However, there are two big advantages of BaseScope over other techniques such as quantitative PCR and RNA sequencing, and I'm sure our listeners are well aware of this. First, both RNAscope and BaseScope have single-cell resolution, and second, even more important for this study, the hybridization is done in conserved tissue, allowing morphological information and an overview of a whole tissue section. This image is a pseudo-overlay of the hybridizations with probes for the 4 ErbB4 isoforms on independent serial sections, since BaseScope works in a single-plex mode. When I compared the results of the hybridizations for the 4 ErbB4 isoforms, I realized very quickly that JMb and CYT-2 are the major ErbB4 isoforms throughout the brain. Nevertheless, I was able to identify areas that exhibit distinct patterns of ErbB4 expressions, and one of these regions is the corpus callosum. The corpus callosum is a white matter area mainly consisting of oligodendrocytes that myelinate the neuronal axons running through this area. BaseScope quantification revealed that JMA and to a lesser extent CYT-1 ErbB4 isoforms are the predominant splice variants expressed in the corpus callosum. Since this was a novel finding, I used quantitative Tachman PCR to measure the 4 ErbB4 isoforms using RNA isolated from carefully microdissected corpus callosal tissue. Both approaches again gave extremely similar results regarding high levels of JMA and CYT-1 transcripts in this area. That supports the fact that when quantified properly, and that will be discussed in the next section, BaseScope is a valid method to quantify relative levels of distinct RNA variants. Interestingly, if one carefully analyzes the distribution of BaseScope signals in positive cells, you can see that there are distinct patterns of expression. While JMA was expressed evenly throughout the corpus callosum in many cells at low levels, there were a few cells that were positive for JMb, but they expressed high expression levels. These differences were also reflected in the quantifications. Based on these observations, which I should stress were only possible by the use of BaseScope and not qPCR, we hypothesized that the JMA and JMb ErbB4 isoforms are expressed in distinct cell types. To test this hypothesis, I began by resorting to the use of multiplex fluorescent in situ hybridization RNAscope. ErbB4 was expressed by GABAergic neurons, here in green, and oligodendrocytes in magenta in the corpus callosum. ErbB4 positive oligodendrocytes were numerous and expressed low levels of ErbB4. On the other hand, I only detected a few GABAergic neurons, but they had high expression levels of ErbB4. Ultimately, to show that GABAergic neurons and oligodendrocytes are the two cell populations expressing JMA and JMb ErbB4 transcripts, I developed a new protocol combining BaseScope and immunostaining. This allowed to analyze splice variants in sections of transgenic cell type-specific GFP mice. Here, I am showing NG2 GFP mice, a marker for oligodendrocyte precursor cells. While JMA ErbB4 transcript overlaps nicely with GFP-positive cells, JMb is absent. In GABAergic interneurons analyzed in sections of gut GFP mice, JMb was highly expressed. JMA transcript was low but not absent. Lastly, to evaluate the relevance of our rodent studies for expression in the human brain, which could be relevant to the differences in ErbB4 isoforms reported in postmortem brains of schizophrenia patients, I analyzed ErbB4 splice variants in microdissected human brain tissue by quantitative real-time PCR. As you can see on this slide, we found that JMb and CYT-2 comprise 70 to 80% of ErbB4 transcript in the cortex, whereas in the corpus callosum, JMA is present in about 70% of ErbB4 transcripts, and the CYT isoforms were expressed at more or less equal levels. The relative distribution of ErbB4 transcripts are very similar between the mouse and human brain, suggesting that the cell type-specific expression of ErbB4 transcripts in the adult human and mouse brain is conserved. In summary of this first part, I hope to have convinced you that BaseScope is a highly sensitive and specific assay to analyze low abundance splice variants. Moreover, quantitative analysis of BaseScope is comparable to standard quantitative methods such as Tagman qPCR. Using BaseScope, we identified regional differences in expression of ErbB4 splice variants in the mouse brain that are conserved in the human adult brain and originate from regional differences of cell type-specific ErbB4 splicing. GABAergic interneurons mainly express JMb and CYT-2 isoforms, oligodendrocytes and oligodendrocyte precursor cells mainly express JMA and CYT1 variants. These findings are important because the relative expression levels of JMA and CYT1 ErbB4 isoforms are augmented in the postmortem prefrontal cortical regions of schizophrenia patients, suggesting a possible implication of these cell types in the disease. Now I would like to come to the second part of my talk that focuses on the quantification of in situ hybridization signal using a custom-made CellProfiler pipeline. I would like to begin by showing you how the pipeline works using an RNAscope example, then lay out how I am setting up a quantitative experiment, and finally explain how to use CellProfiler and adjust a pipeline. As Mohammed indicated, there are commercial softwares available to specifically analyze in situ hybridization signals, such as Hello, Spot Studio, and Aperio, and more generic image analysis software like Imaris. The principle of these analysis tools are very similar, and you can see two examples here on the right. The area of nuclei is increased in order to account for somatic localization of RNA, and then the in situ hybridization signals are assigned to these cells. You can set up similar analysis with free open source software such as ImageJ, and I'm going to present you today analysis done with CellProfiler, which is a free model-based software from the Broad Institute. So this is the example. I hybridized a section of mouse somatosensory cortex with 3 RNAscope probes to address in which cell types my gene of interest is expressed. This gene of interest in here is ErbB4, which is shown in magenta and was used as a channel 1 RNAscope probe. As labeled by the yellow arrowheads, ErbB4 expression overlaps with the GAD1 probe that identifies GABAergic interneurons shown in white. But ErbB4 is absent from neurons that are identified by the glutamatergic excitatory neuron probe vGlut1 that appears here in green. It is important to emphasize that this image is a maximum intense projection of a C-stack and it was taken at 63x magnification. The reason this is important is because at lower magnification, the puncta corresponding to the hybridized transcripts can appear as fused dots. For the successful use of the quantification pipeline, it is important to have single dot resolution. For more information regarding the in situ hybridization methodology and quantification methods discussed here today, you can also consult a manuscript that we are currently preparing for current protocols of neuroscience and hopefully will be published early next year. When using the pipeline, you upload your images that, if not already pre-adjusted in another program such as ImageJ, should be converted to grayscale and separated into single-channel images. Next, if necessary, the in situ hybridization signal can be enhanced to help to identify signal over background. The next steps are the heart of the pipeline. It's about the signal recognition that is performed by a model called Identify Primary Objects. As you can see in the images and the diagram on the right, individual objects are recognized by this module. Signal is mainly distinguished by an intensity threshold, but objects can be also filtered by size. In this process, clumped objects are segmented, which is especially crucial for DAPI-positive nuclei, which is shown here in blue. The size of the DAPI-positive nuclei objects are now increased to account for the somatic localization of mRNA. Then, by a simple overlay, the transcript is assigned or related to these cells. And positive cells are identified by filtering these cells for a minimum of expressed transcript. In this case, I am using, for instance, at least 5 ErbB4 transcripts per cell and 10 to 15 for the 2 cell markers as a cutoff. Now, I'm of course particularly interested in these 4 ErbB4 positive cells, and I would like to understand if these cells are also positive for one of the other markers. To find out if they are double and triple positive cells, the transcripts are related back to the subpopulation of ErbB4 positive cells, and the cells are filtered again. In this case, we obtain 3 cells that are also positive for the inhibitory marker GAT1, and 1 ErbB4 positive cell that also has VCLUD1 transcript. By masking the transcripts back to these cells, per cell expression levels are obtained. And it becomes clear that the GABAergic interneurons express high levels of ErbB4, much higher than this excitatory neuron that only has 6 dots, and they are barely visible down here in the lower left corner. At the end of the pipeline, you can choose to measure object size and intensity, export your images and spreadsheets. So finally, I want to show you some possibilities of how to represent the data. I usually look into transcript levels per area and the percentage of positive cells per all cells. In this cortical example, the excitatory marker VCLUD1 in green is of course expressed at highest levels. Next, I looked into the subpopulation of the ErbB4 positive cells and found there were 4 ErbB4 positive cells, of which 3 were also positive for the inhibitory marker GAT1. Per cell, ErbB4 transcript levels were much higher in these inhibitory neurons compared to the excitatory neuron positive for ErbB4. Lastly, you also obtain information about every single object, which includes position, transcript levels, and if measured, size and intensity. With this information, in this case, I was able to represent the original image and plot the cells with different sizes depending on their expression level. In the next couple of slides, I want to walk you through how I am setting up a quantitative experiment. To allow statistical analysis, you should preferably perform 3 to 4 independent RNAscope or BaseScope samples or animals. In general, you can get along with low sample numbers because you will analyze thousands of cells. So even with low animal numbers, you will obtain robust data. In my example from the somatosensory cortex, I analyzed 2 animals bilaterally and ended up with more than 18,000 analyzed cells. Next, you will need to optimize both your imaging and quantification settings. You may want to do this with small areas, such as a single field of view, to avoid wasting time imaging the whole area and then realize that imaging settings still need to be optimized. Imaging of RNAscope should be always done at high magnification, 40 to 63x for single dot resolution. For BaseScope, 20x is sufficient since the dots are bigger. I image a couple of distinct areas to make sure the pipeline works in different parts of the image. In this case, for instance, I imaged in different cortical layers and also from different samples. The latter is particularly important if you use samples such as wild-type and knockout samples with dramatic differences in expression levels. Then you would start to adjust your pipeline. And depending on the outcome, you would need to go back to the scope and image with different image settings. What I mean by this comment is that fluorescence intensity of a channel might be too high or too low for good signal recognition with the pipeline, in which case you will need to have changed the laser or the gain settings at the microscope. So you go back and forth until you have optimized both the imaging and quantification settings. Then you will image the whole area that you're interested in, which in most cases will require tile scanning at 40 to 63x magnification. I also recommend to image through the whole section in a C-stack. If you only image a single plane, you might introduce a bias or get inconsistent results if you do not image from the same plane between samples. Finally, you will analyze the images with the optimized CellProfiler pipeline. This should be done in the background mode of the program to reduce computational memory. The exported images and tables are then analyzed. Because the program runs in the background mode where you cannot follow its progress, I usually double-check the quality of the analysis with the exported cells. This, for example, are all double-positive red and white cells in the somatosensory cortex, and it looks pretty reasonable. Doing this, you will very quickly see if you had issues with background or edge artifacts. Finally, it's best to perform the experiment for all samples at the same day. But if large sample size precludes this possibility and requires performing the analysis on different days, it is important to randomize different samples, such as comparison of samples from wild-type and knockout mice, because the efficiency of your experiment can vary between days. In this slide, I show an example of the final analysis of the expression of ErbB4 in the somatosensory cortex. This is about the area that I analyzed across 6 cortical layers that are indicated here by the Roman numbers. I analyzed several parameters, but now in the interest of time, I'm just showing an additional possibility of how to represent the data. Using the information about the position of the cells and the transcript itself, I could plot this histogram across the 6 cortical layers. And it becomes evident that most ErbB4 positive GABAergic interneurons are located in deeper cortical layers. In my last few slides, we will discuss how to use CellProfiler and to adjust settings. Again, the program is freely available, and I am running version 3.00 for this example. This is the window that opens when you open the program, and the first thing you will have to do is import the pipeline. That works by a simple drag and drop into the highlighted area on the left. You can use example pipelines from the CellProfiler website or published pipelines, including the ones that we published earlier this year from the homepage of CellProfiler. And of course, you can also build your own pipeline by adding modules. Then, very similar, you load your images, again by a drag and drop into this field. I'm using TIFF files that I pre-adjust in image J to grayscale images and 1 file per channel. Here you need to pay attention how you name your files. If you have a look under the tab Names and Types here, the program recognizes which file is which by the name of the file. In this case, the DAPI channel image is recognized by the name channel 1 C1 anywhere in the name of the file. After you have successfully loaded your images and pipeline, you will start a test mode for adjusting the pipeline. And I do this generally with a couple of different single field of view images from different areas and samples. Make sure you are now in the foreground mode. All windows are shown during the run, and then start the test mode. The window will change a bit. The current model is highlighted here, and the model settings are shown on the right. You can find information about each single setting by clicking on the Help button. The test run is run model by model. To advance a model, you click on the Step button, and then the analysis window of that model will pop up. You can evaluate the analysis and then adjust the model if necessary. I unfortunately do not have the time to explain every single model today, but I want to highlight one of the key models and explain its settings. This is one of the models Identify Primary Objects. It's for the red channel corresponding to the ErbB4 signal. This window will pop up when the model is run. You see on the top input and output image. In the left lower corner, you see which objects were accepted in green and which were rejected in magenta. Since this model is already adjusted and there's very little background in RNAscope, most of the signal is here in green. On the right, you can also find information about how many objects were detected, their size, the intensity threshold that was applied. In this case, I'm satisfied with the analysis of the model, but let's say you're not and you would like to adjust the settings. Then you would run the model again and see if the analysis has improved. So how do these model settings look like? In this model, again, identify primary objects. On the top, you have input file, and output objects. Then the size filters are set. These are in pixel units, so they will vary with the magnification you imaged. I'm usually not very stringent here. This middle part sets thresholding method and intensity. In this case, I'm using the auto algorithm to distinguish between fore and background. And the lower part describes the segmentation of clumped objects, which is not as critical for the in situ hybridization signal, but it's very critical for the nuclei. Here it is done by intensity of the signal. When all your models are adjusted, you would then load and run your whole image set. Again, as an example here, the bilateral cortical analysis in 2 animals. Make sure that you're now on a background mode, hide all windows on run, and then click Analyze Images. Depending on how your pipeline is set up, you will export images and files. As indicated earlier, I used the exported images as a quality control to exclude background issues and edge artifacts. These are the exported CSV files. The most important one is the one that says underline image. It contains all the summary information, like the numbers of identified objects. For each object type, there is also an additional file that has information about each individual object. This is an example for all double positive white and red cells. In these tables, I can find expression levels per cell of the ErbB4 transcript, as well as the localization of the cell. I hope that the second part of the webinar has been useful to explain the use of our custom-made CellProfiler pipeline to quantify your in situ hybridization data. Please feel free to download and use our published CellProfiler pipeline. If you would like to revisit the example or questions to the quantification pipeline remain, please consult either our recent manuscript published in Molecular Neurobiology or our upcoming methods paper on this subject. I would like to thank my supervisor, Andres Bonanno, without whom any of this would have been possible. I also thank current and past lab members involved in my training, my supervisors and committee members at University of Bonn, the group around Emily Park at Advanced Cell Diagnostic for setting up the probes, and I'm also thankful for the support from NIH course and our funding sources. Thanks for attending this webinar. With that, I would like to give back to Christy, who is leading the Q&A session.
Thank you, Ms. Erben. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to give us feedback by taking our exit survey after the webinar. All right, we'll now start the Q&A. Larissa, in slide 9 in your presentation, there seem to be ErbB4 points that appeared to reside outside the cell. Was that an artifact?
I'm not 100% sure. It might also be signal that is sitting in dendrites, and with the technique that I'm using to visualize the GFP after in situ hybridization, the dendrites unfortunately are not visible. So it might be simply just a signal that was transported into the dendrites.
All right. Can you speak to the time frame of the experiment, imaging and quantification, and how long does it take per sample?
So that's, of course, highly variable, depends how big the area of interest is that you want to analyze. So in general, obviously, RNAscope is done in a day, and then the next day you would go to image. The imaging through the C-stack and tiling at 63x or 40x is a little bit time intensive, so it might take about 30 minutes per image. You obviously have a couple of images. The setting up of the pipeline itself also can be time intensive depending on the experience you have. And then the analysis itself is fairly quickly. Again, it depends on the size of the images, but that's running in the background and usually done in about an hour or two.
Okay, great. Does your method distinguish RNAs that could be targeted for degradation? And how does fixation or storage impact signal longevity or stability?
I'm not sure to the first part of question. Maybe Mohammed can say, maybe that question is more applicable for Mohammed from ACD.
Mohammed, do you want to chime in?
Yeah, I missed the first part of the question. What's the question?
Does the method distinguish RNAs that could be targeted for degradation? And how does fixation or storage impact signal longevity or stability?
Oh, I think we've done multiple experiments regarding this matter. I have to transfer this question to our support if anybody is interested in the answer. I think we have a couple of white papers about this. They can get them from them.
All right, great. Mohammed, you mentioned ISH and IHC together. Could you talk about that a little bit more?
Yeah, so this is a new method that we're going to release soon. And this method is shows you compare the RNA with the protein expression, of course. And this method can be automated using Leica biosystems, for example, and Ventana. Of course, yes, we can combine both on IHC and IF on the same section. And we highly recommend the RNA assay first, followed by IHC or immunofluorescence, and using well-validated antibodies for high-expressing protein targets as well. So, and of course, please contact our support team if you want more information about this new assay that we're going to release.
And can the dual ISH-IHC be performed on an automated machine?
Yes. As I mentioned, you can use it through Leica biosystem, Bond RX, or Ventana Discovery ultra-automated scanners.
All right, great. Larissa, should the combination of BaseScope with immunostaining for GFP, is that also possible with RNAscope? And can you also use other antibodies in the immunohistochemistry?
Yeah, so I mean, as Mohammed mentioned, that is a little bit tricky, and the problem is if you combine in situ hybridization with immunohistochemistry, the in situ hybridization requires a protease digestion to let the probes enter the sample, and that's of course contraindicative for the immunostaining when the epitopes might be destroyed. So I tested actually, in fact, a lot of antibodies, and only about 10% are working in my hands. It is possible with other antibodies in the paper that is going to come out next year. I'll mention a couple of different ones, and it is definitely, there's no difference to use it with, if you use it between BaseScope or RNAscope, both it's possible.
Great, thank you. Mohammed, what neuroscience-related probes are available for use with RNAscope?
So the most popular probes are for GPCRS, such as the RD1 and the RD2, since they are notorious for having poor antibodies. In addition, we offer cell markers probes such as RP-FOX3, GFAB, and AIF1 and S100B, as well as markers for glutamatic and GABAergic neurons such as VGLUT1 and VGLUT2. However, ACD can design probes against any target. You just submit your sequence for NCPI accession number to our probes design team.
And what's the difference between the version 1 and version 2 fluorescent assays?
The main difference is that version 1 assay is designed for use with fresh frozen or fixed frozen tissues, whereas the version 2 assay is primarily for FFPE tissues. In addition, both assays can be performed manually, but the V2 assay can also be performed on the Leica automated stainer.
How many targets can you detect simultaneously?
You can detect up to 4 RNA targets simultaneously, and then combine with IHC or immunofluorescence to detect additional protein targets.
And how long does the RNAscope assay take to perform?
So the manual chromogenic RNA assay is about 8 hours, and of course it can be divided up to over 2 days if you prefer. The fluorescence assays vary depending on which version you are using. The version 1 takes as little as 6 hours, and the automated assay on the Ventana or Leica takes about 12 hours, but with only about 30 minutes of hands-on time. And the dual IHC-ish IHC is between 11 and 14 hours, depending on the assay.
Great. And can you detect multiple targets at once with BaseScope?
Yes, you can detect up to 2 targets with BaseScope duplex assays. In addition, you can combine BaseScope-ish with IHC to detect additional protein targets.
And is BaseScope automated?
Yes, the assay can be run on Leica Bond RX or Ventana Discovery automated platform.
And how short of a sequence can you detect with BaseScope?
BaseScope can detect a sequence of 50 or more nucleotides.
All right, thank you very much. Those are all the questions that we have in our queue. We'd like to thank Mohammed Kutaish and Larissa Erben and our sponsor, Advanced Cell Diagnostics. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archive version will be emailed to all attendees. Thank you for joining us for this Genome webinar.